In this part of the lecture, now we're going to look at the effector functions. So we had all of those T cells that went through proliferation and differentiation into these effector cells, right? Well, what are those effector cells? We're going to take a look at their properties and we're going to look at their functions of a handful of them now. If I can advance the slide. There we go. So after their differentiation in the secondary lymphoid tissue, both CD8 positive T cells and CD4 positive T cells are going to travel out to the infected tissue. So they're not going to fight the infection from that secondary lymphoid tissue, but rather that's where they become activated, and that's where they're going to be interacting with the dendritic cells, and then eventually they're going to interact with B cells too, but we'll talk about that in a second. So naive and effector T cells are going to be very different in the way that they now use their T cell receptor or the way that they interact with antigen. Before, for a naive T cell, it needed that 1-2 punch, right? It needed the first signal of antigen specificity, and then it needed the second signal of the co-stimulatory molecule. That secondary or that co-stimulation isn't necessary for effector T cells. They can go out and they could just use their antigen specificity and be stimulated to do the work that they're supposed to do. Now for effector T cells, their target cells are going to be very wide range depending on what their specific effector function is. Naive T cells, they are only going to interact with antigen on dendritic cells, but these effector T cells really can recognize antigen expressed on any MHC class II, and that includes macrophages as well as B cells. And so those are our other antigen presenting cells, or APCs. Taking away that co-stimulatory molecule allows for the T cells, those effector T cells, to do their work much faster and efficiently without having to deal with all the other stuff that's going on in inflammatory environments. So it's pretty nice that that co-stimulatory requirement has been removed. CD8 positive T cells as well do not have to have that co-stimulation once they've become fully activated. And that means that they can go out and they can kill any cell that's going to be infected by a virus. What has to happen is those infected cells just have to be displaying specific antigen in conjunction with MHC class I. And then it will recognize, and it just needs that one signal to say, oh, yep, we're going to kill you. Sorry. It's the name of the game. Okay. So so we did see we needed the co-stimulation for the activation. We needed IL-2 to get proliferation and differentiation to occur, but only the recognition of antigen for the actual effector functions of cytotoxic lymphocytes. So effector T cells then exert their function through a cognate pair or a conjugate pair. And this means that the T cell needs to be connected to the cell that it's interacting with. It might be a B cell, it might be a macrophage, anything that is going to have, in this case for our CD4s, anything that's going to have an MHC class II molecule on the surface. CD4 positive T cells at that synapse then will be able to secrete cytokines directly to the cell that it is in conjunction with, and it will help the cell do certain things depending on what cytokines we're looking at. Effector T cells, effector CD8 positive T cells only have the one function, and that's to kill, and so they have cytotoxins that get delivered directly to the target cell, and this could be any cell of the body that expresses the antigen on MHC class I. Now I have this breakdown with these 5 different cells, and then the cytokines. We're going to look at each one of those individually, so there's no point in me going through them right now on this table. No, you can come back to this table, though, if you want to have a summary of all of them. Refresher, cytokines. Cyto, cell, kine is a chemical, I believe. Is what we're going to go with. Cytokines, they're tiny little proteins that are secreted from one cell to another cell as a message. And they can be membrane bound as well. They don't have to be fully secreted. But they are going to be the messages that drive the adaptive immune response, and it drive a cell and tell a cell which transcription factors to turn on, which proteins to make, and then those proteins that they make will have some sort of function. So they really truly control all aspects of the immune response when that CD4 positive T cell binds to whatever receptor cell or whatever cell that is going to connect with. Changes in gene transcription is really gene expression is really what happens in that target cell, and then that is what the effect is. Sometimes the cytokines can act in an autocrine fashion, acting back on the cell that secreted it. Sometimes it is paracrine, acting on a nearby cell or a cell that it's attached to. And many of these cells, because they're between a lymphocyte and a macrophage, or 2 lymphocytes, they're called interleukins. So you'll see a lot of these cytokines are actually labeled IL, interleukin 2, interleukin 4, interleukin 21. Okay, but know that interleukins are cytokines. Many of them are made by T cells, and they're going to be used in different combinations to really influence how the immune response moves forward, what it does, what the body does to fight whatever infection, or how it affects the target cell. And so the 5 major effector T helper cells, or I should say just effector T cells, CD4 positive T cells, are T helper 1, T helper 2 cells, T helper 17, T follicular helper cells, those are all the T helper cells, and then we have Tregs. And there are cytokines that are distinguished along or attached to these different cell types. Sometimes they are the cytokines that drive their production, but then they could also be the cytokines that are produced from them. So when we think about T helper 1, we should kind of start to think about inflammation or pro-inflammatory when we think T helper 1. It's a pre-inflammatory response that is provided or that is driven by a T helper 1. And so interleukin 2, interferon gamma, those are going to be 2 kind of like pro-inflammatory type cytokines. T helper 2, we should start to think not as much inflammation, but more antibody, more B cell driven. And so we're going to see IL-4 and IL-5. We'll look at these in detail. T helper 17, not as talked about as much, going to be a lot with like parasites. T follicular helper cells, very important for B cell, driving B cell, and so IL-21 is going to be important there. And then Treg cells, that one is IL-10 and TGF beta. When you hear Treg, you should think anti-inflammatory. And so TGF beta and IL-10 are both anti-inflammatory cytokines. Cytokines are made and used immediately. They're not going to be stored up, and they're not going to be saved for later function. It is use them right when they're hot. So the pathway that drives them, again, sometimes it's the same cytokine that drives it, that they produce. Sometimes it's a different one, and this is a really good summary chart. We'll look at each one of these, break them down individually. So I'm not going to go through this table right now. We'll look at them on the next subsequent slides. But the pathway of this differentiation is going to be determined by the cytokines that are driving in the immediate environment will drive the helper cells to become a certain type of helper cell, but then will in turn drive the production of other cytokines that guide the cells as to their target cells with what they do. So let's look at each one of these individually, starting with T helper 1 cells. So these are all CD4 positive T cells that we're looking at, these helper cells. And think inflammation. When you hear Th1, think inflammation, because these cells are going to produce cytokines that drive or increase inflammation. And so IL-12 is going to be an important cytokine. It's made by dendritic cells. It's made by macrophages that will drive a CD4 positive T cell to become a T helper 1. Interferon gamma is also going to be produced because a T helper 1 cell is going to get an NK cell to produce interferon gamma. Interferon gamma, really major pro-inflammatory cytokine. So then the cytokine production by the T helper 1 cells is going to create an environment that further differentiates T helper or helper T cells into T helper 1. And so we'll call this a polarized pathway. So T helper 1 polarized situation, and more inflammation drives more inflammation. And so very important for bacterial infections, important for intracellular viral infections. We'll see that when we get a little bit later on, but yeah, so pro-inflammatory cytokines with T helper 1. Really important function then for a T helper 1 cell, because we're looking at a bacterial infection, is to get macrophages to be more efficient at phagocytosis. So be better at uptaking pathogen and be better at killing it once it's been uptaken. And so when the T helper 1 cells arrive to infected tissue, there's going to be macrophages that are there that were part of the innate immune response, because they're resident tissue cells that are going to be the first ones there. There'll probably be neutrophils there because they were called up. But these macrophages have been doing phagocytosis. That means they've been taking in pathogen, they've been processing it, and then they're going to be displaying it on MHC class 2 because they are antigen presenting cells. The T cell receptor of this now mature activated effector T cell can recognize the antigen in conjunction with MHC class 2, and then go ahead and start producing the pro-inflammatory cytokines that will drive macrophages to become better at killing. So let's look at, it's called macrophage activation. Here's an image that shows how that interaction occurs. You'll notice there's not a co-stimulatory molecule here because there isn't one needed, because this is already an activated T cell. So just antigen has to be recognized by the T cell receptor, and MHC class 2 has to be recognized by the co-receptor CD4. And then we do have CD40 ligand binding CD40, but because we're looking at a macrophage, it's going to induce the macrophage to become better at killing. So it can change gene expression within the macrophage to become more microbicidal. It'll start to kill things it takes in a little bit better. And so that intravesicular bacteria are killed by the activated macrophage, and hopefully that then will produce a situation where there's not a whole lot of extra tissue damage, and the macrophage can kill what it needs to kill and just be efficient at it. Interferon gamma is also going to be that driving cytokine. Okay, so T helper 1, think pro-inflammatory. T helper 2, not very inflammatory. It's not anti-inflammatory, it's just not as inflammatory. It is really great against parasites. They really do not generate a lot of inflammation, but rather promote repair, recovery of damaged tissue where parasites might have been causing an infection. And so T helper 2 cells are going to drive B cells to produce IgE. So it will interact with B cells to get isotype switching to occur. So it's more of a humoral response. IgE has an Fc epsilon portion that is taken up by mast cells, basophils, eosinophils. And then when that IgE on the surface of those mast cells, basophils, and eosinophils gets cross-linked with a pathogen or a parasite in this case, then it becomes activated to degranulate, and then those granules can harm the parasite. A crucial cytokine for a T helper 2 then is interleukin 4. And so this will turn on transcription of interleukin 4 and interleukin 5, which further promotes switching of classes to IgE, making antibody, and so not a very inflammatory situation. Okay. For both T helper 1 and T helper 2, the cytokines that drive their differentiation, so interferon gamma for T helper 1, IL-4 for T helper 2, not only did they drive the production of those effector cells, but they're also produced and secreted by the effector cells. And so you get a positive feedback situation. The more interferon gamma is going to drive more T helper 1, which will produce more interferon gamma, and so on and so on. And so we call this polarized. I used that word a few slides back, but you can have a T helper 1 polarized situation or a T helper 2 polarized situation. When you have a T helper 1 polarized situation, you have more inflammation and you tend to see cell-mediated immunity. It's dominated by the T cells. The T cells are doing the work, and they're getting macrophages to be better at the work they do, but it's all really T cell driven. On the other side, a T helper 2 response is getting B cells to respond and make IgE. And so you see antibody being the dominant effector protein here. And so this is called humoral immunity. So we have these two arms of the immune system, the cell-mediated and the humoral. Cell-mediated is going to be T cell driven. Humoral is going to be antibody driven. Okay, we're going to look at T helper 17 and T follicular helper cells together on the same slide, starting with T helper 17. And we talk about it some. It is going to be a T cell that secretes IL-17. So that's nice that the number matches what the interleukin is. And it's inducing stromal cells, epithelial cells to secrete cytokines that bring neutrophils into the tissue. And then you'll see this in gut mucosa, but not a whole lot that we talk about with T helper 17. Probably more discussed is T follicular helper cells, because they are very important in interacting with B cells. In fact, that's their job. These effector T cells don't ever leave the secondary lymphoid tissue once they find their soulmate B cell. So they will attach with a naive B cell presenting antigen on the surface of MHC class 2, and will be induced by interleukin 6. Then T follicular helper cells are going to secrete cytokines that will drive B cells to switch antibody into the best affinity antibody, as well as the best class for whatever type of infection is occurring. So these T follicular helper cells are going to move from that T cell area where they were initially activated by dendritic cells, and they're going to move into a close area called the B cell area. where all the B cells are hanging out. They'll interact with naive B cells, and this time they're going to be checking for antigen specificity on the surface of MHC class II on B cells. They are activated already. They do not need the co-stimulation of B7, but the B cells have not been activated yet. They're still naive and they're hoping to find their soulmate T cell. So the B cell receptor is going to be binding antigen and processing it, internalizing it, processing it, and then going to be presenting on MHC class II. The T follicular helper cell is going to see the antigen peptide on MHC class II, and if it matches and it has the same ligand for that peptide, then that means that they're going to form a synapse. Keep in mind, is it on here? I'm not sure where I have this. It might be here, it might be another slide. Keep in mind that B cell receptors can recognize unprocessed antigen. And it could be carbohydrate, could be protein, could be something else. T cells can only recognize processed antigen, and it has to be a peptide. So even though a B cell is going to be presenting a peptide that a certain T cell will recognize, it doesn't mean that the B cell receptor and the T cell receptor have the same antigen specificity. Okay. So the two cells then will form a synapse, and also binding at that synapse is CD40 and CD40 ligand. When CD40 and CD40 ligand bind, then that's going to tell the T follicular helper cell to produce cytokines that will drive the production of higher affinity isotype switched antibody. So it starts that B cell maturation process to go ahead and switch out those IgMs, those IgD constant regions. Go ahead and make some IgGs, IgAs, secrete that stuff and get it out there. And then that B cell becomes mature, activated. The T cell is an effector cell, and the B cell will then differentiate into a plasma cell, memory cell. But we'll look at that in a later chapter. Oh, here's the slide that I was talking about where, yeah, the antigen doesn't necessarily need to be the exact same, like the specificity, but it has to be linked. It's a linked recognition because it comes from the same antigen. It's just the B cell will process it so that the T cell can recognize it. So B cell and T cell epitopes, although derived from the same antigen, don't necessarily have to be the same macromolecule, or will they be the same macromolecule? So, okay, Treg cells. This is a totally different situation. Treg cells, or regulatory T cells, they are going to be there to really keep things in check, to keep the immune response under control. And once a pathogen has been pretty much cleared up, the T helper or the Treg cell will start to shut down the immune system once, that pathogen no longer poses a major threat. It will facilitate in tissue repair, tissue healing, and sometimes these cells are called FOXP3 positive cells or CD5 or CD25 positive cells because they have CD25 on their surface, and FOXP3 is the transcription factor that is specific to them. So if you ever see that, know we're talking about a Treg. So Treg cells make cytokines that are immunosuppressive or anti-inflammatory in nature. Two major ones are IL-10 and TGF-beta. They are going to be anti-inflammatory or prevent inflammation from occurring. Suppression really does depend, though, on physical contact between a Treg and its target cells, so that synapse still has to form. And Treg cells are going to enter the secondary lymphoid tissue that is where the response is being generated, where those other effector T cells are located, interact with dendritic cells, and then prevent the dendritic cells from getting more T cells activated. And so this is going to be in direct contact with these T cells with the dendritic cells. So those are the 5 major types of CD4 positive T cells. We're going to wrap up this chapter looking at just a few slides of information about CD8 positive T cells, because they also are a big, huge component of cell-mediated immunity, because they are going to kill any cells that are infected by pathogen. Once like a virus, for example, gets inside of a cell, antibodies aren't going to be able to do anything. An antibody can't neutralize a virus that's already infecting a cell. And so the only way to effectively remove that virus is to kill the cell that it's living in. So effector CD8 positive T cells, or cytotoxic lymphocytes, are going to contain cytotoxins. They make them, they store them in lytic granules, and then they will deliver those little granules directly to the infected T cell through a synapse. They contain a couple different enzymes, granzymes, porphyrin, proteoglycans that are going to be destructive to the cell, rupture the membrane and different things like that, maybe cause apoptosis. But this killing mechanism that CD8 positive T cells use is very much like what we saw with NK cells and the lytic granules that they used to kill altered cells or virally infected cells. So NK cells and CD8 positive T cells, very similar in their effector functions. Well, their effector function, because it's just to kill. That's the natural killer cell, the cytotoxic lymphocyte. They are there to kill cells. It's just that the CD8 positive T cells are going to be there when the innate immune system is completely overrun and the NK cells just can't keep up anymore. After a CD8 positive T cell has been activated by specific antigen, and it gets the co-stimulatory signal from a dendritic cell, then it can start to synthesize these cytotoxins. It keeps them in an inactive form so that they don't destroy themselves, but packages them into granules. And then once those granules are deposited directly to the target cell through the synapse, that's when they become lytic and end up causing destruction to the target cell. It does happen at very close contact. You don't want this stuff spraying all over the interstitial tissue, right? You're just hanging all the interstitial fluid and then just like, killing things here and there. So it is a very direct target, a direct delivery of those granules. So it occurs at the synapse. Those CD8 positive T cells are going to go right down the line, and usually you're going to have like an epithelial, a line of epithelial cells that are infected, and they will examine what is being expressed by the epithelial cell on MHC class 1. If it is the pathogen being displayed on MHC class 1, then the cell that is displaying it will end up being killed. And then the T cell will move on to the next one and check and kill, and then move on to the next one and check and kill. And so one single CD8 positive T cell can actually kill a lot of infected T cell or infected target cells, just one after the other after the other. So the apoptosis is going to be the major way that these cells are going to die, programmed cell death. And essentially what happens is the chromatin of the nucleus just breaks down. And once that happens, then the cell is going to die. And so it really is killing it from within. And it's not going to be super disruptive to the other tissues. It's going to kind of condense in on itself and then get cleaned up by macrophages. And so it is good that it doesn't, explode everywhere and then release viral particles out to go infect other cells. So it does prevent the release of those infective pathogens to nearby cells. Besides cytotoxic function, though, CD8 positive T cells are also going to contribute to the immune response by secreting cytokines. And CD8 positive T cells love inflammation, and so they're going to be shooting out that interferon gamma. Interferon gamma will inhibit, or at least slightly inhibit, viruses from replicating or getting into infected cells. They will increase the processing of viral antigen. They will, interferon gamma will increase the presentation of viral antigen by target cells. And then interferon gamma, which we've seen multiple times so far this semester, is that it will activate macrophages at being better at phagocytosis. And so they can clean up the tissue of dead cells a little bit better. We made it. There's just one more slide here, and it's a big summary slide of everything that we looked at, where we have a differentiation of the T cells, whether it's CD4, CD8 kind of, mostly CD4, where they're going to interact and become activated in the secondary lymphoid tissue. Upon that, CD8 positive T cells, along with Th1, Th2, Th17, and Treg cells, are going to leave the secondary lymphoid tissue and go out and do their effector functions. CD4 T follicular helper cells are going to be maintained in the secondary lymphoid tissue, but they're going to move to the B cell area and interact with B cells and get them to secrete antibodies. That's their effector function. So all these effector functions then are going to be predicated on the fact that the T cells have to be activated by a specific peptide that's been presented on MHC class 1 by dendritic cells. Well, I guess the effector ones, the effector T cells, can recognize MHC class 1 on other cells as well, and MHC class 2 on B cells and macrophages. CD8 positive T cell effector function is to kill infected T cells. T helper 1, 2, 17, and follicular helper cells are going to help other cells do the work that they need to do. And Treg cells are going to try to shut things down, prevent an overrun of the immune system. Okay, we made it. We made it through cell-mediated immunity activation. So we'll stop there, and I guess we'll pick up with humoral immunity next.